Hi guys, welcome back to my channel. Thanks for such good response on my previous videos. Keep supporting my channel. This is an educational channel. Don't forget to like, share and subscribe to my channel. Do support my channel. So today's topic is on for biology for engineers. So let us begin with the presentation. The title and the contents of this presentation is Introduction to Enzymes, Enzyme Separate Substract Specificity, Enzyme Catalysis, Enzyme Regulation, Enzyme Kinetics, Enzyme Cofactor, Enzyme Denaturation, Enzymes in Industry, and Enzymes in Medicine. So first let us get into the introduction to enzymes. Enzymes are biological catalysts that speed up chemical reactions in living organisms. So they're highly specific, meaning that only catalyst one type of chemical reaction. Enzymes are made up of proteins that hold into specific species shapes to carry out their functions. So they are made up of proteins and they hold into specific shapes to carry out their functions. Whereas they are also highly specific, they can only catalyze one type of reaction at a time and they speed up the chemical reactions in the living organisms. So here is a diagram of substrates and active site of enzyme, how they catalyze the chemical reaction. Now let us study the enzyme substrate specificity. Enzymes have active sites where substrates bind and undergo chemical reaction. So enzymes have their own active sites. Only in those active sites the substrates bind and undergo chemical reaction. The enzyme substrate complex is highly specific, ensuring that only a correct substrate can bind to the enzyme. Enzymes can undergo induced fit where the active site changes shape to accommodate the substrate. Now enzymes catalysis. So enzymes lower the activation energy required for chemical reaction to occur. So these enzymes work is to lower the activation energy required for chemical reaction to occur. They do not alter the equilibrium of the reaction, but they can increase the rate of which equilibrium is reached. So enzymes can catalyze both catabolic and anabolic reactions. Catabolic reactions are reactions which break down, whereas the anabolic reactions which build up. The enzymes are proteins that act as a biological catalyst. They lower the activation energy which required for the chemical reaction to occur and they speed up the chemical reaction that do not change by the reaction. Catalysts are those which speed up the chemical reaction and are not changed by any type of reaction. Now let us learn the enzyme regulation. The enzyme activity can be regulated by the factors such as temperature, pH, substrate concentration, etc. So these competitive inhibitors bind to the active sites of an enzyme, preventing the substrate from binding. Non-competitive inhibitors bind to the site other than the active site, changing the shape of the enzyme and reducing its activity. Now with the graphs we can see that the factors which regulate the enzyme activity are temperature, pH and substrate concentration, whereas the temperature in the temperature enzyme activity graph, with the increase in the temperature, the enzyme activity also increases, reaches a certain level, and suddenly reduces um, to zero, where suddenly the enzyme activity reduces. Whereas in the pH versus enzyme activity graph, it gradually increases the the enzyme activity gradually increases with the increase in the pH and reaches a point, stops there and reducing. The enzyme activity starts reducing once it reaches certain pH level. Whereas it is not the same with the substrate concentration. It is zero, the enzyme activity is zero or null until the substrate concentration reaches a certain point. After that, it starts increasing gradually and reaches a certain level. After reaching a certain level, it will not decrease nor increase, remains the same average. It maintains a uniform line with respect to the substrate concentration. So here is a graph of the enzyme regulation. So now let us learn about the enzyme kinetics. Enzyme kinetics studies the rate at which enzyme catalysis reaction occur. The Michaelis-Menten equation describes the relation between the substrate concentration and reaction rate. Enzyme inhibitors can affect the kinetics of the enzyme catalyst reaction. The next one is enzyme cofactor. Some enzymes require cofactors such as metal ions, or coenzymes to function properly. So cofactors can assist in enzyme substrate binding or participate in the chemical reaction. So we already learned what are the enzyme substrate. So these cofactors assist them in the enzyme substrate binding and participate in chemical reactions. Enzymes that require cofactors are called hollow enzymes. So there are few enzymes which require cofactors for the binding and participate in the chemical reaction. Those enzymes are called hollow enzymes. Now let us learn about enzyme denaturation. Enzymes can be denatured by extreme temperatures of pH levels, causing them to lose their function. In few previous slides, we learned the effect of pH and temperature on enzyme regulation. Now we can learn how these temperature and pH level affect the denaturation of these enzymes. So causing them, they lose their functionality. Denaturing disrupts the enzyme's shape, leading to loss of substrate binding and catalytic activity. Denatured enzymes can sometimes be renatured if the condition are returned to normal. So we know how to renature these enzymes which are already denatured. We can do that by returning it to the normal condition. Now let us know what are the application of these enzymes. These enzymes are used in industries. They are used such as food production, pharmaceuticals, and detergents. They improve the efficiency of chemical processes and reduce the need for harsh chemicals. So enzyme engineering allows for the modification of enzymes to enhance their properties of specific applications. We can learn the use of enzymes in industries. Basically, food industry, the dairy products uses are lactose, protease, catalase, and rennet are the few enzymes used in dairy production. Baking are maltogenic amylase, glucooxidase, and pentosinase are the enzymes used in baking. Wine and fruit juice are pectinase and beta glucanase. Protease and peptide are used in meat. Maybe they're used for preservations also. And the second application of enzymes is in medicine. Enzymes play a crucial role in various metabolic pathways and cellular processes in the human body. We usually know how enzymes act in our day-to-day metabolic activities, whereas how we can learn how these enzyme deficiencies can lead to genetic disorders such as phenylketonuria or lactose intolerance. We have usually come we know few people who have lactose intolerance, and this is due to what enzyme deficiencies. Enzyme replacement therapy is used to treat the enzyme deficiency by providing the missing enzyme to the patient. So we also know how to cure it. We can conclude by saying that enzymes have various applications in their day-to-day life, whereas it is very important for our metabolic functionality of the human body, and there are many uses in the industries also. These are essential for biological molecules and regulate chemical reactions in human organisms. They continue to focus on research for their potential applications in various fields. These are the uses and the properties of enzymes. Thank you.